Good day, everyone. Thank you for attending today's webinar on culturing Apical-Out Organoids with Xeno's free Vitrogel system, a part of our webinar series on Vitrogel hydrogel for organoids. I am Christopher Oak from the Well Bioscience, and I will be your moderator alongside our speakers for today, Dr. Kahara Mendictaweli from the Well Bioscience, and we'll be presenting first, and afterwards, Chloe Harris from the Cardiff University. During this webinar, you can utilize the Q&A tab on your left side for any questions you have. We will collect and present them during the Q&A session afterwards. We will have some quick poll segments during the webinar. Please feel free to participate as we go. Also, the webinar will be recorded, which can be accessed later for your convenience. It is my pleasure to introduce Dr. Kahara Mendictaweli from the Well Bioscience. Dr. Kahara earned his doctorate from the Texas Tech University, where he investigated the mechanism linking adipose renin angiotensin system to inflammation and ER stress. He did his postdoctoral research at Rutgers University, where he explored the novel role of intestinal fatty acid binding proteins in regulating glucose metabolism and energy hemostasis through specific ligand binding using numerous models, including 3D organoids. He is currently with the Well Bioscience as a research scientist too, overseeing the 3D organoid modeling unit, primarily focusing on developing and refining various Vitrogel hydrogel systems to support organoid cultures. I'm going to turn it over now to our first speakers, Dr. Kahara. You can go ahead and present. Thank you.
Thank you, Chris, for the introduction. Let me share my screen. Hello, everyone. Thank you once again for joining today's webinar, where we will be discussing how to generate apical-out organoids using Xeno-free hydrogel system. So first of all, I would like to pay your attention on the key takeaways of today's webinar. First, Vitrogel system is pure synthetic alternative for animal-based ECMs like Matrigel and BMC. And also, this system is room temperature stable, meaning that you really don't have to use ice or 4 °C when you carry out your experiment. And also, Vitrogel organoids supports apical-out polarity in contrast to apical-in polarity we observe with animal-based hydrogels. The last key takeaway is this system also supports long-term organoid growth, actually over 60 days, where you see a very steady growth over time. So with that, I would like to briefly introduce organoids. They are three-dimensional, tiny self-organized tissue structures. More importantly, organoids mimic both functional as well as structural features of organs. Now, these 3D structures are becoming extremely popular among research communities, mainly due to their physiological similarities to the intact tissue. Now, in this graph, it shows the number of publications related to organoid research published over the last 20 years. And we can clearly see that that number has significantly increased, I would say, last 5 to 6 years. And this trend is going to increase more. Ever since the breakthrough study conducted by Handclaver's group back in 2009, where they first successfully developed intestinal organoids using LGR5 positive stem cells, many other organoid models have been developed and established for numerous tissues and systems, including intestine, liver, kidney, breast, brain, and many other tumor lines. So due to these beneficial features and advancements in research, organoid cultures can be considered as a preferable model in many in-depth biological studies. So there are several applications as I listed here. This is a very good model for drug discovery and screening studies, disease modeling, personalized medicine, host-microbiome interaction, co-culture, and also nutrients and drug absorption related studies. This would be an ideal model. Now, while organoids closely mimic in vivo features or the in vivo environment, for certain areas, it still raises a question whether these organoids are the best model, especially due to the polarity of these organoids. So I would like to switch gears here and quickly introduce the polarity of organoids. which refers to the asymmetric distribution of cellular components and functions. Now, for instance, if you take enterocytes as an example, which are found in the epithelial layer of both small and large intestine, these enterocytes exhibit a distinct apico-basal polarity, meaning they have a specialized apical domain or the membrane with the microvilli and special functions. They also have a basolateral membrane which interacts with the underlying tissue and blood vessels. So as you can see here. So the maintaining proper polarity is crucial for development as well as functions of these cells and organoids. Now, based on the organoid polarity, actually we can identify two main types, apical-in organoids and apical-out organoids. As the diagram displays here, in apical-in organoids, the apical domain, this part is actually facing the center or the lumen. Now in these 3D structures, you can't really access this area. However, when you look at apical-out organoids, this apical domain is facing outside, which can have a direct interact with the ECM or the media, whatever the treatments that you do. Now, interestingly, when you culture organoids in animal-based hydrogels like Matrigel, BME, Caltrex, like animal-based hydrogels, they support apical-in type of polarity. And these are some data we generated intestinal organoids cultured in Matrigel. And we use phyloidine, which is an apical marker. And we can clearly see that these organoids show apical-in polarity. Now, as I briefly explained the importance of apical domain in the previous slide, they have specialized functions like absorption, it could be nutrient or drugs, and sensory transduction, barrier formation, secretion, as well as host-microbial interaction. So all these specific functions are taking place at this apical site, which ultimately triggers many downstream pathways, which will regulate overall homeostasis of these cells. So now, why do we need apical-out structure? Especially if you are carrying out experiment using an apical-in type of organoid, the direction or the interaction actually taking place from the basolateral side, which is the opposite direction. And it will not really recapitulate the interaction we see in vivo. So unfortunately, this type of apical-in organoids fail to fulfill this key requirement. And this is why sometimes we consider whether this is the correct model for certain type of experiments. Now, however, if you use apical-out type of organoid, you can see that the apical domain is facing outside and it can directly interact, and the direction would really mimic in vivo. So here I have some data showing apical-out organoids support fatty acid absorption. Now these first three images shows apical-in organoids or basal-out organoids, and these three are here apical-out. And both these were treated with fluorescent fatty acid C1 BODP C12, which represented in green color here. And we can clearly see that how apical-out organoid supported the absorption or the fatty acid absorption compared to apical-in, where you see very small absorption. So these results indicate how important it is to select organoids with the correct polarity for your experiment, because it will have a huge impact on your final results. So with that, I would like to quickly mention some other applications of apical-out organoids. This is a good model for gastrointestinal studies like nutrient uptake, drug absorption, metabolism, host-microbial interaction. Also, you can study viral infection, mucus secretion, cilial motility in terms of respiratory studies. But these apical-out organoids can also be used for drug screening, toxicity assays, personalized medicine, as well as disease modeling type of experiments. You can use these apical-out organoids. So with that, let's see how you can generate apical-out organoids. This is the current process, actually how to use apical-in organoids cultured in animal-based hydrogels and then convert into apical-out organoids. So first you do you culture organoids in the hydrogel. This is somewhere around 5 to 7 days. Now it is very important to remember you have to use undifferentiated organoids in this situation, because if you pass that stage and if organoids start differentiating, when you flip the polarity, they will lose viability. So it is important to make sure that you use undifferentiated organoids. And then you, once you have those organoids cultured for 5 to 7 days, you dislodge organoid and matrix and transfer into a vial to perform depolymerization of matrix, so basically to get rid of all the gel. And based on the protocol published in Nature protocols like recently, they suggest incubating for an hour in a solution include with EDTA PBS. You can actually use a similar solution. Again, now this step is also very crucial, because if you incubate 1 hour at 4 degrees or little longer, it will start affecting the viability of these organoids. And also, if you take it too soon, what will happen if some gels are remaining, then the conversion won't be successful, because then you will have some mixed polarity. So once you depolymerize the matrix, you transfer all these organoids and resuspend in organoid culture media, and then you transfer into a low attachment plate. Usually within first 24 hours, this flipping starts, but this process would take somewhere around 2 to 3 days to here in the suspension culture to generate like successful apical-out organoids. Now you can see, as I just mentioned, it takes few days, and this process has several limitations. So first one is it reduces viability, and higher loss is also seen, especially if you start with differentiated. If your organoids are already differentiated, then you will have a lot of like dead organoids during this process. You need to identify undifferentiated organoids, and for this you may need some level of expertise to see what stage these organoids are. And also when you culture organoids, they are not at the same stage. Some organoids might grow faster and start differentiating compared to others, so there are some high variability. Another limitation is you will see incomplete polarity flipping, basically mixed polarity. Now, this is mainly due to the depolymerization of matrix. If you don't get rid of all the gel, you will have this type of issue during this conversion. And also, the culture process usually carried out in suspension culture that will not support long-term organoid growth, because we know that the matrix actually will support all these mechanical strength and bio cues to make sure that these organoids are maintained properly. However, in suspension cultures, that cues will not be there, so you won't be able to carry out long-term cultures in this stage. Last but not least, it is time-consuming and also requires some expertise. So this is where we the Vitrogel comes to the picture, and we introduce Vitrogel organoid, which can be used to solve most of these limitations. Vitrogel organoid naturally supports apical-out morphology. You really don't have to do any of these changes, flipping, because the organoids and the tissue fragments, you just have to culture in Vitrogel organoid, and it will naturally support apical-out morphology. And not just that, Vitrogel organoid system can be used to massage and expand your organoid lines, and also it supports long-term organoid culturing. So here I would like to quickly go over some other benefits of Vitrogel organoid. As I mentioned, it is room temperature stable, easy to handle, and Xeno-free, 100% synthetic hydrogel. Now, compared to most of these animal-based hydrogels, those are tumor-derived. So there are like thousands of unknown components which might directly or indirectly affect the growth of organoids as well as results. And there is a high variability, batch variability in those animal-based gels. So we don't have that issue, and it's a key for clinical applications. And it supports apical-out polarity, and many different types of organoids can be cultured using Vitrogel organoid, and very easy to harvest, as well as this is automation friendly. So now let's see how we culture organoids using Vitrogel organoid system. So the process is very straightforward. First, you get a pellet of cells or tissue fragments or organoid fragments, and then you mix it with organoid culture media and make a suspension. Then mix the Vitrogel organoid with cell suspension at 2:1 ratio. Basically, 1 ml of Vitrogel organoid can mix with 500 microliters of organoid suspension. And you do a quick mixing and then plate it, and you can incubate for 10 to 15 minutes at room temperature. Now, this is important. People who are familiar with using animal-based gels know that this initial incubation we usually do at 37 degrees inside the incubator. But the principle behind cross-linking and gelation is different in Vitrogel. So you can actually leave it in the hood and incubate it for like 15 minutes at room temperature, and it will start like soft gel formation will begin. So after this incubation, you can cover the gel with additional media, which will further increase gelation, and then you can place in the incubator for maintaining these organoids. So that's the process. With that, I would like to go over some results we generated using organoids cultured in Vitrogel system. First, we'll see how Vitrogel organoid supports apical-out polarity. Now, the data here, we show mouse intestinal organoids cultured in Vitrogel system. The staining green is phalloidine, which stains actin, which is an apical locator, and DAPI is a nuclear marker. Now, we can see these young organoids, as in the diagram shows, clear apical-out polarity is seen in these organoids cultured in Vitrogel. Now, these are some mature organoids cultured in Vitrogel system and Matrigel, and we can clearly see that the apical-out polarity is maintained in these Vitrogel cultured organoids compared to apical-in polarity we observe in organoids cultured in Matrigel. Now, the other important factor I would like to emphasize here is Vitrogel organoids generate lumen-like structures we usually see in the intestine while maintaining this apical-out polarity. Now, when you forcefully flip polarity of apical-in organoids, what they will do, they will make a very dense structure, and they won't produce this type of lumen structures. So this is an important key benefit of culturing in Vitrogel. Now here we have some mature intestinal organoid cultured in Vitrogel system over 50 days. And these structures are big, and the videos here represents part of the organoid. And the staining was conducted to confirm both structural and morphological features of these organoids. And the green color represents SOHO1, which is a tight junction protein expressed in intestine epithelium. So this protein is crucial for forming barrier and regulates permeability of the intestinal wall. And we see a very clear expression of SOHO1 in these mature organoids. And the red color represents beta-catenin. Again, it's a very important protein, especially Wnt and beta-catenin signaling pathway is essential for intestinal homeostasis as well as regulating organ regeneration. Now, beta-catenin is also considered as a basal membrane locator. So the positive expression of both these SOHO1 and beta-catenin in these mature organoids is a clear indicator of healthy growth over time in Vitrogel system. All right, so now we know that the intestine is a complex organ. It has many different types of functions, and all these complex functions are actually regulated by specialized cells. So next, we wanted to confirm when you culture organoids in Vitrogel system, whether it will generate all these specialized cells. So here I'm showing just one due to the time. The red color MUC2 stains goblet cells, and we can clearly see that these mature organoids cultured in Vitrogel generate these goblet cells while maintaining their apical-out structure. And the control we use is the Matrigel. And when we culture organoids, we see that goblet cell formation, but you can clearly see that Matrigel organoids maintain their apical-in polarity compared to apical-out polarity in Vitrogel. I think some more details will be covered by Chloe during her part, the second part of today's webinar. And a few more slides. Now, here, the data here shows how Vitrogel supports long-term organoid culture. These are intestinal organoid cultured over 60 days. And we can clearly see that these organoids actually, not just they were able to remain 60 days in the Vitrogel organoid system, but they were also able to maintain these physiological and morphological structures. But when we look at in Matrigel, we use the same set of cells cultured in Matrigel. Usually what we see within first 2 weeks, organoids grow pretty fast and they start making this dense structure, and they reach their expansion limit and then they burst, eventually die. So you can't really go beyond, I would say, 30 to 40 days. So Vitrogel systems support long-term organoid growth. And while Vitrogel supports long-term culturing, organoid can be easily passaged using Vitrogel organoid system. This is the workflow for that. Once you culture them, I would say within first 24 to 48 hours, you will start seeing this clear edge structure. This is a good indication to say organoids are viable. And then when you maintain on maintenance media for about 4 to 6 days, you will start seeing clear growth, increasing size. And by the time day 10 to 12, these organoids will be ready for passaging. Now, you might see a little different compared to other animal-based gels, the rate of the growth rate. But what we see is a very steady growth over time. And if you, based on your requirement, you can also passage organoids at this stage. But if you wait for 10 days, your yield will be much higher. So these results show stable passage within Vitrogel organoid system, where we use the same organoid line and passage several times. Now, the key thing that I would like to emphasize here is, throughout all these passages, these organoids were able to maintain their morphological and structural features. Now, for a fact, we know that when you passage several times in animal-based gels, you will lose some of those morphological and structural features. But in Vitrogel system, you can maintain those structural and morphological features, which is important. Additionally, organoids which were cultured in Vitrogel system can be easily passaged into any other hydrogel system. So these results here demonstrate organoids cultured in Vitrogel system and then passaged into Matrigel. And we can see how these organoids were kind of like survived initial transfer, and then you see a very steady growth over time. All right, so with that, I would like to summarize a key few points about Vitrogel. Vitrogel organoid is Xeno-free synthetic hydrogel. It also supports wide range of organoids and can be used to culture long-term organoid culturing. It naturally supports apical-out polarity, and also you can do stable passaging within Vitrogel system as well as across different other hydrogels. So with that, I would like to thank you everyone once again for joining today's webinar, and I'll pass it to Chris for the second part. Thank you.
Thank you, Dr. Kahala, for your great discussion on how the Xeno-free Vitrogel organoid hydrogel support apical-out and its advantages and the ability for long-term organoid growth. Very excited to introduce our next speaker, Chloe Harris, who was funded by the NC3Rs, is a PhD candidate in cancer immunology at Cardiff University. Her research focuses on replacing mouse immunotherapy models with human colorectal cancer organoid T cell co-cultures, aiming to develop targeted cancer therapies while sparing healthy tissues. Using patient-derived organoids and advancing sequencing techniques, she has also worked to reduce animal-derived products in research, such as Matrigel. Chloe holds a Master of Science with a first-class honors from the University of Bristol and previously worked as a scientist at Advanced Instruments. I want to welcome Chloe. I turn it over to you.
Thank you for coming, and thanks for the invitation to today's webinar. I'm Chloe, and I'm a second-year PhD student at the University of Cardiff. I'm as part of the Gallimore-Godkin Lab, and today I'll be speaking to you about my PhD project, which is centered around the replacement of mouse models in colorectal cancer and the immunotherapies using patient-derived organoids. And as part of this project, I'm also looking to replace Matrigel in the system with an animal-free alternative as a further reduction technology. And today I'll be sharing with you my work using Vitrogel organoid formulations. So this is a background into the whole project, which is founded by the organoid biobank Stephanie in our lab has previously set up, which includes organoids from both tumour and healthy adjacent tissue from patients undergoing surgical intervention, which we then dissociate and make organoids from for both the healthy and tumour tissue. And then we also store the crypts as well. As well as this, from the same patient, we also then isolate PBMC from blood sample and store this, then with the main aim to then combine all of these components in an organoid T-cell co-culture system where we aim to enhance tumour-specific CD8 T-cell mediated killing while sparing the non-malignant organoids in a fully autologous system. So delving into the organoid biobank further, we have currently selected 8 patients of pairs of organoids which have been sent off for both RNA and whole genome sequencing to identify any potential T-cell restricted targets from either neoepitope prediction or differential gene expression. The table at the top here really just shows the heterogeneity of our patient cohort that we have so far, highlighting the patient and disease characteristics. And one of the main things that we're focused on is the microsatellite status of these patients. And if you look at the tissue slices here from 2 of the patients in this cohort, you can see a nice difference between microsatellite unstable, which has a high immune infiltrate, and microsatellite stable, which would be classified as immune cold. If you look over to the MSI tumour here, you can see that the immune infiltrate, which are all the nicely multicoloured dots, infiltrating into the tumour, which is in purple. Whereas if you look over to the MSS tumour, there are immune infiltrate here, but they're restricted to the stroma and they're excluded from the tumour. And this is something that we're looking to enhance in our co-culture studies, as it's known that MSI tumours often respond better to immunotherapies in comparison to MSS. So as I've mentioned, the main aim of this study is to determine whether using the information from our biobank, can we specifically target CD8 T cells to only target tumour organoids to create an autologous system to test immunotherapies on using the patient-derived organoids and the patient PBMC. This is a previous video from an experiment in our lab showing T cells navigating through a matrix in order to get to the organoids and kill them. So you can see the organoids are nicely here in yellow, and over time the T cells in blue migrate through the gel and dock onto the T cells, dock onto the organoids, in order to shrink and kill the organoids over time. And then this is something that we want to recreate in this whole patient-specific system. Another important tool that our lab has developed for organoid studies is OSCAR. This is an organoid cell enumeration machine learning pipeline, which we sought the need to develop. As we all know, organoids are 3D structures and grow differently in a patient-dependent manner, making it nearly impossible to standardize and determine the number of cells within a well of organoids. This is something that we thought was crucial for determining target to effector ratios in our co-culture studies. Therefore, OSCAR was born, and this is an organoid segmentation and cell number approximation using regression tool. And this only requires a bright field image of your organoids. And here's just a quick overview of how this is made. but I will spare the technicalities for now. So far, the tool has been trained on countless organoid images and validated using unseen colorectal and pancreatic organoids, displaying robustness without the need for dissociation, with a high degree of accuracy at around 85%. This work is currently under review to be published, but if anyone would like to test out the tool, do not hesitate to contact us, as it would be great to see this tool in action in a variety of different organoid types. So why are we interested in removing Matrigel from the study with an animal-free alternative? And as my work is funded by the NC3Rs, which is the National Centre for the Replacement, Refinement and Reduction of Animal Research, it is encouraged to endorse the 3Rs wherever possible, and not only by using the amazing replacement technology, which is organoids, which can hopefully reduce the number of mice which are used in these statistics here. We would also like to switch out Matrigel. As we all know, Matrigel is currently the gold standard for organoid culture, but it is derived from mouse sarcoma, with one mouse being used for every 6 ml of Matrigel. So swapping this into an animal-free alternative, not only for the ethical implications, but can also induce less batch-to-batch variation due to its defined chemical composition. And more interestingly for me, the researchers have established that animal matrices like Matrigel and Cultrex in this example can induce unwanted CD4 T cell reactivity to the matrix, which of course would be unfavorable for our immune cell co-culture studies. So for moving away from Matrigel and towards an alternative, I drew up several success criteria for my current application. And these include adaptation of the organoids from Matrigel, successful long-term culturing of the organoids, maintenance of normal intestinal morphology, successful T-cell migration and co-culture through the gel, and finally, can organoids be derived from tissues straight from the alternative? Using this criteria, I'll be discussing my experience with Vitrogel organoid for the rest of the presentation. Following on from this, I used bright field microscopy to track the growth of tumour organoids from all four formulations of Vitrogel organoid and compared to what we currently use in the lab, Matrigel, over multiple passages. To complement this, I also analysed LDH release over several time points. LDH assay is a measure of cytotoxicity and cell stress. LDH is released from the organoids into the surrounding area, which can be sampled over time from the media during media changes and passaging. And finally, I'll be showing my immunohistochemical staining of Vitrogel-grown organoids to ensure the presence of key differentiated markers of the crypt, including goblet cells and Paneth cells. So looking at the growth of the organoids, once transitioned from Matrigel to Vitrogel over the first 10 days, you can see that in all 4 formulations, organoids grow over time, indicating that Vitrogel can sustain organoid growth across all the different 4 formulations in a similar rate to Matrigel in this experiment. For consistency in this experiment, I used Oscar to determine the same cell numbers were seeded into each of the wells to ensure for consistency. After these 10 days, I then went on to passage the organoids. And this was quite a harsh passage, but it was good to see how the organoids still managed to grow over time. And the same case still shows here that over time, all 4 formulations successfully supported organoid growth. So this is a final image from these 2 passages at day 21. But in fact, I did culture these organoids for an extra 2 passages after this, indicating that Vitrogel is sustainable and suitable for long-term culture of colorectal cancer patient-derived organoids. You may have noticed that Matrigel-grown organoids appear larger and often more abundant. And as Kahara mentioned earlier, this cannot always be desirable, as this can lead to uncontrolled growth and death and require higher levels of passaging and more use of reagents, therefore making Vitrogel more manageable and sustaining organoid growth in a more controlled way. So to accompany the bright field images I just showed you, Excitingly, the LDH assay also reveals that LDH levels in organoids cultured in all 4 Vitrogel organoid formulations remain comparable to, and in some cases, lower than Matrigel, indicating that there's no increase in cellular damage or stress in the gel, which is extremely encouraging for my future experimentations, as this meant that the organoids were happy in the gel over a long period of time. So once I knew that the organoids grew well in Vitrogel, the next step was to look into the morphology of the organoids a little bit closer, and I did this by using fluorescent microscopy. And here you can see 4 individual slices across the 4 different formulations, and their corresponding Z-Sac videos underneath, displaying the presence of differentiated crypt markers. So we have goblet cells in red, which is marked by mucin, and Panna cells in white, which is marked by lysozyme. And to my excitement, I could see that all of the cell types were expressed in all of the formulations here, which was great news. But one of the main observations I had here was the strong presence of the phalloidin staining around the outside of the organoids, which is something I've not really seen before, especially in Matrigel cultures. So I went to investigate this further. But for comparison, here are some Matrigel grown organoids from the same patient sample. And you can see that the green phalloidin markers here are mostly on the luminal face, which is the apical side of the organoids, which Kahara explained earlier. This led me to the conclusion that Vitrogel organoids were in fact apical out, and essentially the organoids have turned inside out in this gel. So after this discovery, I went on to investigate the uses of apical-out organoids and how this could implicate certain applications. Following a literature search, I found out that apical organoids are in fact more desirable for certain studies, such as microbiome studies and co-cultures, and absorption and nutrient uptake in the gut, as well as also modeling viral and microbial diseases in airway organoids. Previously, this was achieved by placing organoids in suspension and relieving them from Matrigel, as Kahara also explained really nicely earlier, with some nice visuals. But again, it takes a long time and doesn't always ensure that all the organoids are apical out. And this can also only be done in short-term experiments because of the viability of the organoids in suspension. Another option, which is also used, is the dissociation of the organoids and sacrificing their 3D structure in order to make monolayer experiments. This, however, makes the model less physiologically relevant, as one of the main novelties of organoids is its 3D structures. And this is where I think Vitrogel really falls nicely into place, where it supports organoid growth over a very long time and also supports the apical out structure across multiple passages, whilst also expressing all of the differentiated cell types that I've just shown, as well as keeping it in the 3D structure, as well as also it being really extremely easy to adopt in the lab with its easy methodology. So just to summarize, I've really quickly summarized my PhD project so far. So I hope that I've shown to you that patient-derived organoids are extremely useful resources in cancer research and beyond. However, the gold standard matrix for organoid culture, Matrigel, has many different implications aside from its ethical and murine origin. And Vitrogel organoid offers itself as a synthetic alternative, which is able to provide viable and prolonged organoid growth. And also, Vitrogel organoid induces an apical out morphology, which is not typically observed in Matrigel. And this can be beneficial across multiple different applications without altering the physical integrity of the organoids. So with that, I would just like to thank everyone in my lab and my funders, and of course the World Cancer Bank and the patients, as without any of them, the development of this biobank and the model would not have been possible.
Thank you very much, Chloe, for your presentation. So before we moving on to Q&A session, I would like to quickly walk you through the key takeaways of today's webinar. First, is it okay? As I mentioned, Vitrogel organoid system is a pure synthetic alternative for animal-based hydrogels. And the system is room temperature stable. You don't have to use ISO 4 degrees Celsius to carry out your experiments. And Vitrogel organoid supports apical-out polarity naturally, in contrast to the apical-in polarity we observe with animal-based hydrogels. And the third point is Vitrogel organoid supports long-term organoid growth over 60 days. So with that, I thank everyone once again, and turning over to Chris.
Yes, thank you, Kahara. Can you go to the next slide, please? Before we go to Q&A, one more slides here is to remind everyone that we have more webinars on our website. Well, there are multiple applications using Vitrogel for like ex vivo models, injectable in vivo, and cell invasions. So we urge you to watch this webinar that could be very informative for your research. So the Q&A sessions here, we have a few here getting started. We'll go one by one. But Kahara, I think this was for you, is what are the different types of organoids that can be cultured in Vitrogel organoid hydrogel system? And will the culture process be the same across all organoids, or do we have to optimize it based on the type of organoid?
All right, so for the first part, different part of types of organoids, actually, as I mentioned, wide range of organoids are supported by our system. For instance, gastric organoids, intestinal organoids, like intestinal organoids, and then lung, brain, we know that they support very well. And Vitrogel organoid, we have 4 main types, 1, 2, 3, 4. So I would say out of those 4 types, organoid 3 would be the best solution, which has a which supports wide range of organoids. And we also know that several customers have used different types of cancer organoids culture using Vitrogel system. So that's the first question. Second one was, can you please repeat?
Sure. And will the culture process be the same across all organoids, or do we have to optimize it based on the type of organoid?
So the culturing process would be the same. However, now based on the type of organoid that you use, obviously the media composition you will have to change. And also we recommend using, if you recall, 2-to-1 mixing ratio. This is something that you can actually change and see how well that would support your organoid growth. So again, the process would be the same. Yeah, you don't have to really optimize unless media and ratio that you can optimize.
Great, thank you. Next question is, can we transfer organoids from Matrigel to Vitrogel to get successful apical-out organoids?
Yes, so you can successfully transfer from Matrigel or any type of animal-based hydrogel to produce or get successful organoid cultures. But I would say if you are transferring organoids from Matrigel, it is important to remember to use early passage line. Sometimes what we see when you passage several times in Matrigel type of hydrogel, you will see some morphological and structural changes. So that might not really help the recovery. So it's always recommended to use an early passage when you transfer from Matrigel to Vitrogel type.
Okay. Next question here. We have a few more coming in, but let's go to the next one. How do you harvest organoids cultured in Vitrogel system? Can we use trypsin to harvest organoids from Vitrogel?
So organoids can be easily harvested we have our own organoid recovery solution, which is specifically designed to depolymerize the matrix or the gel. And we can also use this Vitrogel organoid recovery solution to harvest organoids from Vitrogel. The second question was trypsin. So now, as I mentioned, this is not enzyme-based depolymerization. So if you really need to break it down into single cells, I would suggest mixing with 1:1 ratio with trypsin. But for normal harvesting, you can easily harvest cells using our Vitrogel organoid recovery solution.
Another question, this time for you, Kohara, is how efficient is the Vitrogel in allowing the growth of pancreatic cancer organoids? How quickly can they be grown?
All right. So for this question, pancreatic, it may depend, I would say, based on, like, if you are taking fresh tissues and directly transferring, you will see a very good support, and you should be able to get, I think I would say, like 1 to 2 weeks. But we have some published data related to pancreatic cancer. I think we should be able to provide that information, the publication, for you to get clear information, because sorry, I don't have the exact answer for that right now. But I would say if you start with patient-derived tissues, we know that it supports very well, and you will see a very stable growth.
Thank you. How compatible is Vitrogel with downstream immunoassays like Luminex or multiplex RNA extraction for transcriptomics?
Okay, so basically downstream application. So we can, once you culture organoids in Vitrogel system, you can, it supports like the RNA extraction. You can proceed with the whatever the kit that you use, and it is compatible. Yeah, you can use it. Okay. Even for immunoassays, because for instance, we have done a lot of staining. For Luminex, when you say, I'm not sure what that they meant by that, but the cultured organoids, either you can harvest them and perform immunoassays, or either you can carry out some experiment while they are growing in the gel.
I think this next question is going to be either for Chloe or Kohara, for you to either chime in on this one. One question is, your Apical-Out organoids also show palladium staining on their luminance side, so it seems you really have mixed polarity organoids, which is comparing apical and basal lateral effects. How do you explain the mixed polarity?
All right, so I think it's a good question. Now, one thing that I would like to emphasize here, while you see this apical-out polarity outside, when you culture organoids in Vitrogel system, the important thing that we see is they make these structural similarities to in vivo what we see, those lumen structures. But I get it's a great point. I think I don't know whether Chloe has something to add on that part. But the important thing that I would like to mention here, we can argue that this is also mixed polarity, or sometimes we can say mixed polarity when you see part of the organoid is flip and the other part is not converted. But it's an area that we should kind of like discuss more. But I would like to emphasize that these structures, like lumen-like structures, we see in Vitrogel, which is normally not possible with other animal-based gels.
Cool. You have anything you want to chime in on that?
No, not really. I agree with what Kohara said. I know that there are other markers as well for apical other than phalloidin, so I don't know if that's a different thing to look at potentially as well.
How does Vitrogel compare to other hydrogel in terms of biocompatibility and support for stem cell differentiation?
So now we have done several stem cell related research using Vitrogel, very well supported by Vitrogel organoid system. And we have also used like IPS derived organoids, and which when you actually culture them in Vitrogel and when you compare with other animal based gels, it's very comparable, even with the size and the way that they behave. So it's, I would say, yes, it's very comparable.
Does Vitrogel support the necessary biochemical cues to promote kidney differentiation from HPI PSC cells?
For kidney, sorry, I don't have an answer for that, but I know that human iPS cells, they very well can be cultured in our system. But I should be able to please provide your information. Maybe we can get back to you. Our technical support team can get back to you on that.
Yeah. This next question is, Vitrogel, can Vitrogel be used in a mantis-like liquid handling system? And I can actually add that for you. Yes, it can be adapted to all liquid handlers out in the market. There's no special equipment required because the Vitrogel system is not temperature dependent. So you don't have any gelation issue with temperature fluctuations. So it can be easily, moved around in the liquid handler like liquid. So it can be definitely adapted to almost liquid handling system out in the market. So making a high throughput screening or high throughput analysis much easier than the animal-based system. If you like this, give us, reach out to us at support@thewellbio, or just give us on our contact information. We can, walk you through that as well. We do have done it on a simple system like an Opentron easily, and it can be adapted for a larger system like the Hamilton or a Mantis or anything like that. One question for you, Kahara, is another one is, how does apical-out orientation improve accessibility for drug testing and pathogen interaction studies?
Okay, so now as I briefly explained, when you have apical-in type of polarity in your organoids, actually whatever the treatments that you do going from the basolateral side, so this is not the correct direction for certain experiments. So if you can really flip it, the apical domain will be accessible from the outside, and all the most, let's say, microbial host interactions that you are willing to perform can be easily, the direction would be very easy to access that apical domain. And same with drugs. So if you have apical-out organoids, drugs or the microbial treatments that you do will have a direct access to apical domain. So I think that would be the advantage.
A few more questions here. I think we have a couple of last ones. Are there any specific cell types or organoid models that respond better to Vitrogel for apical-out orientation?
Yes. So, like as I mentioned, many different types of organoids are supported by Vitrogel organoid system. But we know, like we have a list of organoids that we can provide. Gastric organoids, lung, brain, and many different types of patient-derived cancer organoid types are supported by this.
This is just one of the next questions pertaining to the liquid handling, but okay, how does apical-out polarity impact organoid-based high-throughput screening assays?
Okay, so now when the apical side is facing outside, for screening would be easier because then you can use different markers which you have in the apical domain. So that would be, I think, one of the best advantages using apical-out organoids for this type of screening.
Okay. One more time, a lot more questions came in, but we have one last question. Could the Vitrogel system support co-culture models with immune or stroma cells for more complex tissue modeling?
Yes. So we can we have some application notes in our website where we cultured gastric organoids with, I think, dendritic cells to show how well Vitrogel system support movement of dendritic cells compared to other animal-based ones. So yeah, it supports. And again, now this one, something that you need to decide based on whether you really need to carry out apical-in or apical-out type of organoids. So based on that, you can decide what type of organoids that you're going to use.
Okay, that was the last question. So we're going to end it up now. Again, thank you, everyone. Thank you, Dr. Hara and Chloe, for your excellent presentation, and everyone for attending the webinar. I urge you to actually contact Chloe for any of her studies and work at the Cardiff. It's very intriguing as well. And for Dr. Kahara as well, for any questions you have on our part of the Vitrogel system on the apical-out organoids, or any other questions of supporting organoid types and usage. Also, please connect on the LinkedIn website and go to our website as well for more various 3D cell culture applications, protocols, and scientific resources and webinars, as we mentioned before. Again, thank you everyone for your attendance. We'll have this webinar recorded, and actually we'll send it out back to everyone for your convenience to watch again. Thank you, everyone. Have a great day.
Thank you.